All right, I'll share here today. My presentation is on advances in cardiac cryopreservation. And here we are going to discuss our advancements across a range of cardiac biosystems, all the way from cardiomyocytes with a characteristic length there of 20 microns, then to engineered heart tissue for our second advancement, and then all the way up to rat hearts at the centimeter scale. All right, first some background on the problem our research is addressing. So the problem here is that cardiac tissue has only a limited storage time at 4 degrees Celsius. So this is a plot showing patient survival following a heart transplant. The conventional method of storage for donor hearts is 4 degrees Celsius. And what these authors are showing here is that these donor hearts stored for longer time, 5+ hours, have about a 10% drop in survival for those patients. So by elongating the preservation time there could be potential impact in that application, as well as impact for donor-to-patient matching. So if you can imagine, if you have more time to store these donor organs, to store these valuable biological tissues, then you can fit the right organ to the right patient. Here I'm showing a size matching example where hearts that were too big or too small had worse outcomes. And then moving into the tissue engineering space, there are many up-and-coming tissue-engineered products in development across various labs. There's a challenge in getting those products from where they're produced and to the actual clinics or commercial entities or patients who would benefit from them. Here I'm showing the example of a myocardial patch, getting that to a patient who's had a heart attack. Our cryopreservation method, or preservation method in general, is needed and lacking for such tissue-engineered products. And our group's general approach here is to use a lower storage temperature. So on the y-axis here is the time equivalent in the fridge at 0 degrees Celsius, and you get exponential benefits on preservation time in the stored state from transitioning it to a lower storage temperature. So think about moving a sample from the fridge to the freezer there. That's from 0 degrees Celsius to minus 20 degrees Celsius. You get like a 5x benefit in preservation time there, and then that exponentially just compounds to where you get on the order of years. 10,000 years is probably a high-end estimate, but nonetheless, in the cryogenic storage regime, years of storage in that state. All right, so you might expect from this that a lot of the research in the field is using that low storage temperature to take advantage of that long preservation time in the stored state. But when I reviewed the literature, that is not the case. Make that go away. What is being shown here is a lot of the publications are involving actually that high sub-zero storage regime. And to get a sense as to why, you can look at this paper here, in which they identified 2 damage modes that are salient for cardiac tissue, one of which is ice and the other which is cracking. I'll just give you a sense of where those occur on this chart here, with ice centered around minus 70 degrees Celsius, and where we're concerned about cracking is a regime of lower temperature there. All right, and as you probably noticed, there's a data point all the way to the right there, so a recent effort to use low cryogenic storage temperature for heart despite these damage modes. And you might be wondering who these bold group of researchers are. And this is the research group I decided to join about 5.5 years ago. And the reason they're pursuing this low cryogenic storage temperature is because they have a strategy that counters both of those damage modes. So first, they use the approach of vitrification to counter the damage mode of ice. And then second, and this was the pioneering approach, they use the strategy of nanowarming to counter the damage mode of cracking. And in their publication, they showed that they could effectively eliminate and mitigate both of those damage modes. All right, so what I want to focus on here, though, is a problem with specifically the vitrification aspect of this approach, which is that to avoid ice, to vitrify, you need to replace about half of intracellular water with CPA, with cryoprotective agent, and that can be damaging. So what's being shown here is a model cell on the left. This would not vitrify. If you tried to cryogenically cool it down, it would form ice. But if instead you load that cell with cryoprotective agent, you load it with CPA, then you can get into a position where that cell can vitrify. And I want to clarify here that in this schematic, it's not like the water is somehow separate from the CPAs in the cell. In reality, in an actual cell, an actual organ, these would all be mixed together in solution. And this is important, right, because if you think about how water would form ice, those water molecules would need to rearrange into an ice crystalline structure, and that interaction with the CPA molecules would inhibit that. I'll talk more about cryogenic cooling and rewarming later, but for now, all you need to know is that this high CPA concentration is good for avoiding ice, but there's also a toxicity challenge. So this is a study we did in collaboration with Dr. Namsrai and Dr. Finger's group, and what we found was that there is a toxicity limit on rat hearts exposed to CPA using our methods. And what is being shown here is we want that tissue after this viability staining to look like this fresh control, that dark red color, but instead, past a certain toxicity limit, there's that deathly pale color instead. And this is quite important because this is the level of CPA concentration we need approximately to avoid ice formation. So we really want to raise the toxicity limit above this so we can use that level of CPA in the heart. And to do that, what we decided to do was develop a better CPA solution. And for this, we used the more controlled test bed of cells. All right, so the work in this first part of the talk here, this first advance, is done in collaboration with Professor Singh with his lab. And we use two general types of cells for this study. The first is human dermal fibroblasts, so the original source for this would be human skin. And the second type is cardiomyocytes. And we actually differentiated and cultured these in labs. The original source there is hiPSCs. That's an acronym for human-induced pluripotent stem cells. And to give people in the audience a sense of what this test bed is like, we can compare our system of these hiPSC cardiomyocytes in lab to the different, though analogous, process of human development. So in human development, right, cardiomyocytes also differentiate from stem cells. And the gene expression of these has been analyzed. And if we compare the gene expression, I'm just going to make a rough analogy here to where our cells in lab for our first experiment, our day 13 cells, that's 13 days post-differentiation, are analogous to those of cardiomyocytes in the early embryo, as shown below. And then our cells for day 21 would be analogous to cardiomyocytes in the mid-embryo, as shown below. All right, so to design a CPA screening experiment, what we took was a model-guided approach as follows. So if you look at the model on the left, there would be two key concentration gradients that happen if a cell with no CPA is exposed to CPA around it. One would be the water concentration, which would cause water to go out. And because water transport is faster than CPA transport, that water leaving would cause a transient shrink. And then as the CPA goes in along its concentration gradient, that cell would swell up back up to near its original cell volume. And a common way for simulating this in the field is to use equations with two parameters, one parameter for that water permeability and a second parameter for the CPA permeability. And this is a potential challenge for designing a good CPA comparison because these CPA permeabilities can range all the way up in our study to a value of 7 for furamide. And to give a sense of why that matters, I'm going to use two simulation inputs here. One is a single-step CPA exposure. The single step there refers to the black curve, the CPA curve, which goes from 0 CPA to the full CPA solution, then back down for the remainder. And then this is in comparison to the stepwise CPA exposure, in which the CPA is loaded and unloaded stepwise. So we're using diluted CPA concentrations for those intermediate steps. The red curve there is not super important for this study, though we just chose a temperature profile that might be good for us later in our cryopreservation protocol. 4 degrees Celsius is often used in an effort to reduce CPA toxicity. And then the higher exposure temperature at the end there is to help with unloading, make CPA unloading go a bit faster. All right, so what are the results? So when we simulated the cell volume, we found that the cell volume would dip under predicted limits. That's the red dotted line there, especially in the case of DMSO due to its low permeability. So we're favoring the right profile there, which has the predicted cell volume within those limits. All right, so that's one method's choice, but overall I would say that our early experiments were not super well designed here. Though there is still some information from them, which I'll get to here. So we tested various CPA formulations on the left here. DMSO plus EG, PG, BG there means 2,3-butanediol. All of these are established CPAs in the field. And what I'm showing in the middle here are some of our results for specifically the cell type of human dermal fibroblasts. What you can see from the green channel images there are that the cells treated with the BG CPA had much reduced live staining, indicating lower than healthy metabolic activity. And then if you look at the red channel images, cells treated with DMSO plus BG had increased number of that dead stain, indicating worse membrane integrity. Here's a summary of those results. Thank you to Ahmed for helping with the quantification. And essentially what we found here was that the only comparison that was statistically significant is the one I'm highlighting in the middle there for our viability assessment. This is somewhat interesting because one might expect a priori that the day 13 or day 21 cardiomyocytes would actually be a little bit less robust. I mean, these are not cell line type cells, but at least for CPA, they were quite tolerant to all the CPAs we tested according to this assay. All right, well, why was I saying that this is not super well designed? I mean, it seems pretty clear. DMSO plus BG is bad, the rest are good. Well, the problem comes here, where we tested each of these CPA formulations at the same weight by volume % concentration. Using the same concentration is a common methods choice in the field, but here's the problem with that, which is that different CPAs have different ice avoidance capabilities at the same CPA concentration. This is demonstrated in this table here, which is taken from literature values, in which at the same CPA concentration, PG has about twice the ability to vitrify as formamide as given by that beta parameter there. And so really what should be done is testing each CPA at the desired CPA concentration, which in a cryopreservation protocol, really you just want to load the cell to the minimum you need to avoid ice. You wouldn't want to go much beyond that. And so really that's what we should have done in setting up this study. And so I'm just putting up what our CPA concentrations probably should have been below for our application of trying to avoid ice. This study was rather conclusive for the first two CPA formulations for application because we tested them at 55 weight by volume %, and that's about what we would need to avoid ice, and they were good at those. But for BG, sure, it was bad at 55, but what about 45? That's what we would actually use in a cryopreservation protocol, and we don't know, so our result is inconclusive there. And for formamide, by this viability test, our result is also inconclusive because sure, it was viable at 55, but what about 60? That's how much you would actually need in a CPA exposure protocol for cryopreservation. And so really, the only way for this to be conclusive for formamide is that if formamide was so bad that by some other assessment, it was even worse than these other CPAs in a comparison that was favorable to it. And this is exactly what we found. So formamide had worse recovery of beating in these cardiomyocytes. So this is the same experimental design, tested at 55 weight by volume % for each CPA. And what we found for formamide, both these videos are playing, but the trend was towards much reduced beating, just even the presence of beating altogether, when these cells were treated with formamide. That's summarized in the plot there. That red color indicates that no cells in the video in any of the quadrants of the videos we took recovered beating. And then if we take the subset of cells that were beating, draw regions of interest over that. So that's another comparison favorable to formamide. Okay, the cells that did recover beating, how good were they? And they were slightly worse in terms of the contraction speed than these other CPA formulations. And so from this, our conclusion is that formamide is worse than these other established CPAs. So we can, in fact, draw that X over the DMSO plus formamide column. And this is poised to be a real advancement from what we were doing before. So that prior study I showed in the prior slide from rat hearts, we were using formamide as well as a couple other CPAs in that study, which had the negative CPA toxicity outcome at the CPA concentration we would use for cryopreservation. And so can we do better than that is the question. And to be sure we can do better than that, we tested VS55, which is that CPA formulation we used before, an established one, versus a CPA formulation we algorithmically designed, DEP, and we algorithmically designed this to have a similar predicted CPA concentration to avoid ice here. So that's the bottom number there. So this experiment was much better designed. We can take the result from this and kind of directly apply it into a cryopreservation protocol. And what we found was similar to the formamide result. There was a trend towards VS55 treated cells having worse recovery of beating, We're attributing that to the presence of formamide in VS55 because of our prior work there. And one thing to note is that this red bar is rather high, in part because this time we're using 25 minutes, which is a more relevant final loading step time for the application of cardiac tissue. And then similar to before, if we take the subset of beating cells and analyze those, we found a drop in recovery for VS55 versus DEP. All right, so to sum up this part of the talk, first, to sum up this part of the talk, I'm going to give some principles of good experimental design for CPA comparison. Here's some of what I have learned over the years. First, to get a more interpretable result, you want to avoid osmotic damage for all CPAs. Second, to be able to detect damage, it's important to have some assays in our case, to assess function. If we had only done that initial cell viability assay that can really detect toxicity but may not function, we would not have detected the damage due to formamide, so it was super important we also did this assessment. And then finally, to test each CPA at the minimum CPA concentration to avoid ice, and you can predict that with some literature values, or if you want to go more in depth, you can analyze that yourself. So this is my recommendation to future researchers to have each of these three principles in mind when designing these CPA comparison experiments. All right, and one of our key results here is that DEP is better than VS55. That's that conventional CPA formulation used in the field. And so what we're looking to do here is take that improvement, DEP, and apply that to some systems that are in need of a cryopreservation method, such as heart tissue. And this is the second part of the talk, the largest part of the talk. And engineered heart tissue, or EHT, is a rather straightforward system for us to target because it's generated from the same cells, the day 21 cardiomyocytes, that we did our CPA screening and development in. This is work done in collaboration with Dr. Lin and Professor Ogle's lab. Dr. Lin did the cardiomyocyte differentiation culture as well as the EHT generation culture for this project here. And essentially what he did is he took those day 21 cardiomyocytes and he seeded them into the PDMS mold divot, as shown on the bottom left there, along with some fibroblasts and some fibrinogen and thrombin to form an EHT, which is essentially cardiomyocytes within a fibrin extracellular matrix. And we did the cryopreservation experiments after allowing these EHTs to compact for about 10 days. So these EHTs contain cardiomyocytes that are 31 days post-differentiation, or day 31. And here's why we're interested in cryopreserving EHT. So if you think about the limit that would occur if you could not preserve engineered heart tissue or other tissue engineered products, you would really only be able to distribute them throughout like a narrow circle there if there was no preservation method. If you had, say, a short preservation method like 4 degrees Celsius, that would give you maybe about a day, as indicated in a prior publication. And that would give you maybe an ability to distribute that to some drug companies, to some biomedical researchers. But the ideal is to have a cryopreservation method, where you could have long-term storage, months of storage, and this would allow you wide geographic access to a lot of different pharmaceutical companies and so on for shipping the product. And this is viewed as a super important element of actually commercializing engineered heart tissue as a biomedical product. All right, and this work is impactful because EHT has not been cryopreserved. In fact, very few cardiac tissue samples have been cryopreserved. Here's a brief literature review of that. So since the discovery of cryoprotective agent, there is one brief report on the preservation of frozen embryonic hearts in which they reported recovery of beating. And then for the decades since, a lot of the reports have been on success using isolated cardiomyocytes, or in 2010, cardiomyocytes differentiated from stem cells. And only recently has there been a report of successful cryopreservation, recovery of beating using day 20 cardiac organoids. All right, so our objective here is to cryopreserve day 31 engineered heart tissue. And when I started this project with Professor Singh, he was talking to me about it. He was just like, okay, this is going to be great. You're going to get success with the EHT, and then we're going to apply this to a mouse model, and then it'll be this big Nature Medicine paper, and it'll be great. And then I was thinking about this, and I was like, well, I'm not sure we'll get past step one, because if you look at the timeline here, we got embryonic, neonatal, less than day 30, day 20, and then this rather ominous statement, which is that the viability of greater than day 30 cardiomyocytes decreases substantially after freezing. It seems like the more mature these cardiomyocytes get, the more sensitive to stressors they are. But CPA stressor is a different type of thing than an ice formation type stressor, so we tried the experiment, even though it was, day 31 cardiac tissue, maybe on the old side. And what we found was that these engineered heart tissues can recover beating following the CPA exposure. So here is the untreated control, no CPA for that engineered heart tissue. And on the right there is beating following the CPA exposure. And you can get a sense to here that the EHT is able to recover its primary function after the CPA, which gave us a good deal of hope in pursuing our method for EHT. And what we did, which was different than the prior work in the field, was we applied an ice-free vitrification approach to cryopreserve EHT. So the only other publication in the literature on cryopreserving EHT used a freezing or ice-allowed approach. And what they found was no recovery of whole EHT beating. They found some cells that were recovering beating, but overall the entire tissue is not recovering beating. Whereas in our approach here, we're able to achieve vitrification, ice avoidance, and so we're avoiding that big damage mode. And before I get into our test of this hypothesis that we'll get recovery of beating here, I want to give some more information on the vitrification approach we are using. And one of the key things to understand here is that using a faster cooling rate favors, it allows you to use a lower CPA concentration. So here I'm going to compare two cases. One is a case of slower cooling using vapor nitrogen, and the second is using liquid nitrogen. And here's our prediction for exactly how slow or fast these are. What you can see is the black liquid nitrogen curve has a faster cooling rate there. If you just look at the x equals 0 part of that plot, which corresponds to the plane of the EHT far away from the mesh, liquid nitrogen causes about a 2x faster cooling rate in these EHTs. And here is how one can make a prediction of the CPA concentration we would need to use. These data were collected and analyzed by Dr. Gangwar in our group. And the way to understand these data is that if you look at the data point on the right, this was done for DEP. So DEP has a critical cooling rate of 0.5 degrees Celsius per minute, which means that if you cool at a rate above that, you avoid ice. And if you cool at a rate below that, you get ice. But then if you dilute DEP down, so you're using less CPA at 85%, then the critical cooling rate goes up, because then you need to use a faster cooling rate to avoid ice. And so what we did here to get a sense of how much CPA concentration we need to use, we made a fit similar to how others have done in the field. We took our estimate for liquid nitrogen cooling rate here, and are predicting about 75% CPA concentration we need to avoid ice there. And for vapor nitrogen cooling rate, we're predicting a slight increase. We would need to use about 3% more CPA concentration in that case. All right, so we need to equilibrate EHTs to at least 75% CPA concentration. And the way we load EHTs with CPA, so these are relatively thick tissues. The way we load them is just by diffusion. So we did the CPA loading in the same environment that was used to generate EHTs. So these EHTs are in the bottom of a PDMS mold divot, as shown there. And we just pipetted the CPA solution on top. And here's what we predict to happen over time. Well, here I'm just going to show you two limiting cases, one of which is 5 minutes CPA exposure, and the other is 25 minutes CPA exposure in black. And here's what we expect to happen throughout the position of that EHT. So at x equals 0, that's the boundary of the CPA solution. For both cases, there's a high amount of CPA loading there. But for 5 minutes CPA exposure, you can see a much steeper drop-off. That region of the EHT farther from the CPA solution is not getting adequately loaded. And for our approach of vitrification, this is what we're trying to avoid. So we're going with 25 minutes, not 5 minutes here, hence our CPA exposure profile, which has a decent safety margin. And this has allowed us to achieve vitrification in all of our samples. All right, so returning to this hypothesis, which is that avoiding ice in EHTs will allow us to go above that literature milestone and recover whole tissue EHT beating. And in our first experiments, this is not what we found. So we did actually get recovery of some whole tissue beating. You might be able to tell that from the video there. But it was at a level much lower than the CPA plus handling control. So some success. We were able to get whole EHT beating using this method, but not full success and kind of a mysterious not full success because it's lower than our CPA plus handling control. Here's our force data. This is collected and analyzed by Dr. Lin, and what is being shown here is that the cryo treatment group had significantly lower force recovery than the CPA plus handling group. All right, so how can this be? I mean, we avoided the ice damage mode during cryogenic cooling and storage. How is this so much worse than our control? Well, here's one thing to know about our protocol, which is that ice during rewarming sometimes occurred in EHTs, specifically when the EHTs were thicker. And one of the ways we assess this is as shown on the left there, where the EHTs during that plunge had that visible white coloration in the case of ice and did not have that white coloration in the case of no ice. This is not super surprising, so if we think about what we would predict for the minimum CPA concentration, we would predict that as the thickness increases, shown on the right there, the minimum CPA concentration in the EHT would decline. And on the top there, I'm plotting what we would expect the desired CPA concentration to be to avoid ice during rewarming. That's a much higher threshold to meet than avoiding ice during cooling. This is consistent with other work in the field. And so it's not too surprising that as we got to a thicker sample, even using that 25 minutes of CPA exposure was not enough to equilibrate the EHTs to a level where we'd avoid ice during rewarming. And in fact, we saw ice in about half of our trials using our thicker EHTs. All right, so this leads one to the hypothesis that ice during rewarming is a major source of damage. But here's why we didn't actually end up pursuing that hypothesis much, which is that when we analyzed our data, we didn't detect a big difference between the EHTs that formed ice during rewarming and the ones that didn't. So here what I'm showing is our recovery of EHTs over time after treatment. So initially, those EHTs had a contraction rating of 2. They were all beating. And then after our treatment, poor Weyhan's EHTs were struggling a decent amount due to our protocol, and then eventually recovered to the level of faint or asynchronous beating. So part of the EHT would be beating at maybe a different BPM than the other, that sort of thing, at 7 days. And that was similar for the case of ice and no ice. And this result is similar for force assessment as well. When we analyze these by ice versus no ice, we didn't detect a difference. So is ice during rewarming a major source of damage? This evidence indicates no. Now, is it a source of damage? I would believe that. But is this the major thing that is causing our results to be so much worse than CPA plus handling? This data indicates that it's not. All right, well, what else could it be? So I would direct your attention to this plot I showed earlier, in which there are 2 key damage modes during cryopreservation, ice and thermal strain. We're avoiding ice during our vitrification approach. But what about the thermal strain damage mode? Is that somehow relevant here? All right, so first some background on thermal strain. You might have also heard of this. This is similar to thermal expansion. Thermal strain would occur in various materials during cooling and rewarming. So if you rewarm EHT, it goes from an initial thickness to a larger thickness. And one can express this change in shed thickness using this equation. So thermal strain would be the change in thickness over the initial thickness there. And the question here would be, does high thermal strain damage EHTs? Well, we know that it damages EHTs by at least one mechanism, which is macroscopic cracking, because we actually found that in some of our early trials. So all the data I've presented thus far has actually been using vapor nitrogen, not liquid nitrogen, because we got macroscopic cracking using liquid nitrogen, so we shifted away from that element of the protocol quite quickly. So using vapor nitrogen, most of the time, 17 of 18 trials, we didn't get any macroscopic cracking, any visible cracking there. But I don't even have any good pictures to show you for the liquid nitrogen case because the EHT wasn't even intact. So there might be a crack all the way through the middle and then a crack through each loop. All right, though, going back to that equation for thermal strain, you might be thinking, wouldn't that actually be pretty similar in both cases? Because if you think about the bulk EHT, it's cooling down to a similar temperature. Wouldn't the the bulk compression be the same and it's rewarming back up to 4 degrees Celsius? Wouldn't the bulk thermal expansion be the same? Why am I attributing this to a thermal strain problem? What I want you to be thinking about is not the whole bulk tissue thermally contracting, expanding, but what happens when you warm up. In this case, I'm showing the comparison during rewarming. When you warm up one region of tissue faster than another, and that region expands differently than the other regions next to it. And so here is our heat transfer simulation result during EHT rewarming here, in which the x equals 1.2 millimeter condition. That's the plane of the EHT close to the mesh boundary. That's where we'd expect the highest rewarming. And this delta T between that plane closest to the mesh and the regions of the EHT which don't have that high of a temperature yet is responsible for the thermal strain that we're concerned about for cracking. And when we think about thermal strain, it's this sort of phenomenon here. All right, how to actually predict how much thermal strain is occurring? Well, what we can do is we can use this equation in which this parameter can be taken from the literature and this from our heat transfer simulation to predict this level of thermal strain for our EHTs. Using our thickness range, it doesn't really matter what the thickness was. It all had about the same predicted thermal strain. How high is this? When you use a predicting an equation from the literature to predict the threshold for cracking, you get that dotted line there. And so we're rather borderline in terms of avoiding cracking during our EHT rewarming. Rewarming is where we really expect a lot of this damage to be occurring because our rewarming is so fast. And so, okay, it's close to cracking, but we didn't actually observe cracking in most of our EHTs. 17 of 18 were good. So really what I'm wondering here is whether there would be a sub-cracking thermal strain damage mode or just some sort of thermal strain damage mode where you don't see it, you don't see that crack under the microscope or taking a picture, but nonetheless thermal strain is causing damage. And so to test this hypothesis, we used 3 different profiles that generated 3 different levels of thermal strain. And here's what we did. So we just simply adjusted the level of liquid nitrogen in our cooling container to give us 3 different cooling profiles. The top dotted line there is all the data I had shown previously. That's using initial temperature of minus 190 degrees Celsius surrounding the EHT. And then we lowered that to come up with 3 different profiles. The one of which, the moderate one there, the blue one, dips slightly below TG. That indicates that this EHT is vitrified, so it would have some thermal strain in the vitrified state. And then the green profile there does not vitrify, so it would have no thermal strain in the vitrified state. And so what we're able to do here is we're able to get a test from minus 130, which would have the lowest thermal strain in the vitrified state, 0, and minus 150, which would have a slight amount, and then minus 190, what we were using before, which would have significantly more. And here are our results for this. So going through these one by one, for the untreated control, we normalized its force to the untreated control for that batch. So the force recovery here is 100% by that, and you can see the visible beating there. And the CPA plus handling, as shown before, it does have this drop in force recovery due to that stressor, though still visible beating. And then for the minus 190 case, there is visible beating here, although the force recovery is significantly lower than that of the CPA plus handling control. And then here is the big test. Can we improve that using minus 150 degrees Celsius? Well, we definitely can improve it. We see is much clearer beating of the EHT there with improved force recovery. And then on the other side, we were thinking, okay, maybe there would be an even bigger improvement here in this EHT that's not vitrifying at all. And actually, there was a drop-off there too. We're not entirely sure why this is, why we found minus 150 to be the best. But here are our explanations here, whereas we think minus 150 might be some good medium, good middle ground between 2 damage modes, one of thermal strain pointing to the right and one of ice pointing to the left. And here's our summary of that result. And so for the force data there, we found no statistically significant difference between the CPA plus handling control and the minus 150 group. whereas we did find a big drop-off when using either minus 130 or minus 190. And this trend was similar in our electrical function assessment. These optical mapping experiments were done in collaboration with the Takachova lab, with Jingming doing the analysis here, similarly finding that trend of minus 150 doing the best over our other tested conditions. All right, so to sum up this part of the presentation, which is our EHT cryopreservation work. First, what we did is we used 25-minute peak CPA exposure to achieve consistent vitrification to avoid ice in our EHTs. Second, to avoid that visible macroscopic cracking, we used vapor nitrogen instead of the more conventional approach of liquid nitrogen during the cooling. And finally, using a intermediate minimum temperature, so not all the way down to minus 190, but a more intermediate temperature, more controlled rate through that glassy region, we were able to get better results, with our best result being minus 150. And then looking forward to the next part of this work, one big area improvement here is now the CPA control. So we got our cryo group rather similar to the CPA plus handling control, but what we can do to do this? What can we do to improve this? And this brings us to the third advancement in this dissertation, which is some work we did in rat hearts. And what was perhaps the most interesting element of our study was the carrier solution screening we did. What is a carrier solution? It is the part of the CPA solution that is not the CPA. So you can think of a vitrification solution as having two parts. One is the CPA formulation, like GEP, and the other is this carrier that is composed of a variety of ions, sugars, buffers, other metabolites. And there are a variety of different carriers used in the field, each of quite different compositions. And the idea is that these differences might matter. So we screened a couple of them. One was the carrier used in our prior work. Another was the carrier used for great success in the kidney. And then finally, we applied these two storage solutions, really 4 degrees Celsius storage solutions, as carrier in our CPA experiments. All right, so these experiments, as with the work in the first part, were just done using CPA exposure only, no cryopreservation. And what we're showing here is that there was no beating in the case of CPA and the Eurocolons carrier, whereas when we switched to using the Celsius carrier, we saw much better results. These videos were taken during normothermic perfusion assessment, so this is after the CPA has been unloaded, and we're hoping to see good beating recovery here. And we got that somewhat inconsistently with the Celsius carrier solution, so what's being shown on the y-axis here is the CPA concentration that the heart was exposed to, and the data points here indicate the level of recovery, so we don't want to see any X's here. And what you can see is that for the Eurocolons carrier solution, it didn't recover beating at the full strength, and then really you can get a sense of how important carrier solution is by how we switched to LM5, and we didn't even get beating at 50% of the CPA concentration we would need to vitrify. And then here's some more information as why we would prefer Celsius to UW. So hearts treated with Celsius spent less time in a rest. That's the rightmost plot. And also they started beating sooner after that normothermic reperfusion. And one of the questions I think that could guide future work here is how can carrier solution have such large effect? Presumably, if we can understand that, we can design these solutions better. We can get to a place with better preservation outcomes and cryopreservation outcomes. All right, so thinking about future directions here, one question I've been interested really since when I joined is why the heart has a much shorter 4 degrees Celsius storage time than the kidney. So what's being shown here is that for the kidney, you can store it about 40 hours prior to transplant, whereas for the heart, as shown earlier in the talk, you really don't want to be storing the heart much more than 5 hours prior to transplant. Why is this? Well, doing a literature review, I came up with kind of two explanations. The first of which was that for the heart transplant case, you need the heart to be beating immediately after. Whereas for the kidney transplant case, it's okay if the kidney doesn't function immediately after. You can have it some recovery time where it doesn't function. So that's one compelling explanation. There's a second explanation, which is that the heart is more sensitive to this stress, more sensitive to injury than the kidney. And this is the one I want to focus on. So here is my explanation. So this is not established knowledge in the field. I don't think there's a good explanation for why the heart is more sensitive to 4 degrees Celsius than the kidney. So this is my hypothesis here, that acidosis is the key reason. So if we take literature data of heart versus kidney at 4 degrees Celsius and look at what happens to the pH, the heart pH declines much more than the kidney pH. And so the question I'm wondering here, which might guide future work, is whether this 4 degrees Celsius metabolism is problematic. Specifically, this sort of glycolytic ATP production and ATP consumption metabolism responsible for that acidosis, is that problematic? All right, and having that in mind for interpreting some of our results here, we found that UW and Celsius were better, right? And there are a variety of differences between these solutions. I'm plotting that all there, but just having that hypothesis in mind of metabolism, one thing that jumps out to me when looking at this is that only for the EC and LM5 groups did we actually give the heart a metabolizable sugar. For the UW and Celsius groups, the ones that did better, we didn't give it a metabolizable sugar. And so I think this is quite interesting. I really hope that someone picks this up in the future and sheds some more light into this. All right, and here is the overall summary of the talk. So we started out with a CPA screening and development in cardiomyocytes, in which the key advance there was that formamide was worse for cardiomyocytes, and then it would encourage others to try out not using formamide in their systems, perhaps using DEP, which is a CPA formulation we developed here. And then second, we got a rather high level of EHT cryopreservation success using our protocol, in which one key feature was to try to limit thermal strain by using an intermediate minimum temperature. And then finally, in the third part of the talk, we discussed the impact of carrier solution and the potential for that sort of finding to guide future work. All right, in my final slide here, I want to thank various people. First, my advisor, Professor Bischoff, with whom I have climbed various summits, both physical, as shown on the left there, and also research summits in this talk. Second, I want to thank Professor Singh, with whom I learned a good deal of cell culture and related techniques. I want to thank Professor Ogle over Zoom, whose support for the EHT work has been instrumental for the level of success shown here. And finally, I want to thank Professor Takachova, who's been with me since my preliminary exam on my committee, and also with our group for our collaborative work on the rat heart project. And I want to thank everyone else who's contributed to this work in ways both large and small. Everyone around here is always so willing to help, and I quite appreciate this. All right, at this point, I thank you for your attention, and I will receive any questions.